All right. You guys got any questions and things? Well, let's wrap this bad boy up. Do a little bit of early embryonic development and click. All right. All right. So the first sort of the stages of embryonic development. I'll just write these up here so we can keep track of them. All right, so when you think about the stages of development, I've listed those on the left there, as we call it, fertilization, first thing that happens, and then the embryo begins to, the cells begin to divide. So on the right-hand side is the title of the stage, right? So on the left-hand side, essentially, is the process, the embryological processes that's happening. And over here is the name of the embryonic stage that we would describe it as. So fertilization, when the egg and the sperm join, produces what is termed a zygote, which would be a one-cell stage embryo. And those cells begin, that cell begins to divide, so we call it cleavage stage or cleavage process. It's just the cells are just divided. And that will form a stage called the blastula, or in human terminology, a lot of times they use the term blastocyst, but same thing. And as the blastula forms, the cells within it are going to start rearranging. So a blastula, as we'll see, is just a sort of a sphere of cells, and they begin to sort of move around and rearrange, and that process is called gastrulation, which produces a stage of the embryo known as a gastrula. And the literal translation of these words, so blast is something that's growing, blastula is a hollow sphere of growing cells, gastrula is to create the stomach. So gastrulation is the process through which the digestive tract sort of originally forms, and that's what's happening during that time period. And then the cells continue to rearrange and fold to form our central nervous system, the primordial central nervous system, which is our nervous system, a neurula. So the process is called neurulation, and this embryonic stage is called a neurula. And at that point, the rest of the organ systems really start to develop. So you have the heart and central vessels, the main vessels of the circulatory system start to form. The kidney starts to form. That really early kidney, that pronephros, and then quickly the rest of the forms, and then the urogenital tract, which comes from the original kidneys. And also the gut is starting to restructure so that the accessory glands like the liver are starting to form. And so we just call that process organogenesis, okay? The eyes are starting to develop, the inner ear is starting to develop. So all of those things are going on. At that point, you could call it an embryo, you could start to call it a fetus. There's an embryonic fetal sort of transition that happens around the end of the first trimester to the second trimester. So these early events through neurulation take place in about the first 21 the first 21 days or so, 3 weeks, 3 or 4 weeks. All right, so fertilization is sperm reaching the egg and fusing and binding to the egg. A lot of sperm will enter the reproductive tract. Most of them, right, are not going to make it all the way through the reproductive tract until they end up actually being able to come in contact with the egg. There are a couple of things that you can think about. There's 2 fallopian tubes, and so a sperm could go into either tube, but there's usually an egg and only 1 tube. So right off the bat, like 50% of them went the wrong way. And the egg is, or the cells around the egg are known to have chemicals that they have on them or that secreted from them that do seem to act as a short-range attractant. They're not going to go long distances, but as sperm start to get close to the egg, they are activated to more intensely swim. So that means as they get closer, they start to swim faster, which means they're more likely to get to the egg. As the sperm move through the reproductive tract, they are moving through an environment that starts modifying what's on the surface of the sperm. And that process is called capacitation, where the sperm will actually interact with an outer layer of cells that are surrounding the egg and release enzymes that they were storing in their acrosome. I mentioned the other day that the sperm really only has kind of a few pieces. It's got the nucleus and the acrosome, and that's really the only 2 main things it has. The mitochondria are attached to that neck piece or mid piece and the flagellum, and they're powering the flagellum, but they're not used for anything in the process of fertilization. So with fertilization, that vesicle called the acrosome is what's going to be pretty important. The sperm actually interact with a protein on the extracellular matrix surrounding the egg. So the egg is going to have this zone around it, which we can see on this slide. So the actual egg cell is here in the middle, and as it was forming, there is an extracellular matrix of glycoproteins that have been laid out around it. And there are no cells there. And when it was studied, it sort of looked like a little clear zone. So it became known as the clear zone, zona pellucida, the zone you can see through. That's what that means in Latin. So this zona pellucida has to be penetrated by the sperm. And this is where it's going to take multiple sperm. The acrosome reaction occurs, and those enzymes that are released are able to sort of make these cells that are around the egg, called granulosa cells or coronal cells, it makes them detach from each other so that the sperm cells can actually push through them. But when it gets to the zona pellucida, it actually digests that glycoproteins so that the sperm can penetrate it. So one sperm won't release enough enzymes to do the whole process. So it takes multiple sperm, probably somewhere around 1,000 sperm, to have enough enzyme released to be able to let at least a few sperm make it all the way through the layer of cells, and then one sperm, the first one through, would then be able to react with the membrane on the egg. There's a specific receptor called the sperm binding receptor that is on the egg. When that happens, the egg will undergo a couple of rapid changes. One that is very rapid, it's a depolarization, and one that takes just a few seconds. The outer membrane of the egg, all underneath it, is loaded with these little granules, little vesicles that contain a glycoprotein, much like this glycoprotein that was already there. And when the sperm fuse, those granules release the contents into that space. And that will prevent other sperm from being able to contact and make contact with the membrane of the egg. This is called the block to polyspermy. Okay? So if more than one sperm fertilized the egg, that would be known as polyspermy. And that's bad news. All right, so what is the sperm bringing to the game? What's it carrying of consequence? Chromosomes, right? So if two sperms fertilize the egg, there'd be a whole extra set of chromosomes that you don't want. So you just want the two sets. You want the set of chromosomes that the egg had, and you want a set from a sperm cell, but only one sperm cell. So you have to prevent multiple sperm from fertilizing the egg, like is what's being shown here. That's not what they're trying to show. They're trying to show like this was one sperm and it went all the way through this process. But if more than one sperm does fuse with the egg, you would have a whole extra set of chromosomes that would make what we would call a triploid situation or a tetraploid situation or pentaploid, how many sperm bound. So the egg needs to prevent that from happening. So the very first step in preventing that from happening is a depolarization. Okay, so the oocyte membrane will depolarize. So normally cells have a polarity where you're looking at the current or the voltage from the outside to the inside, and it's usually around minus 70 to minus 90 electron volts. Well, when it depolarizes, it flips that polarity, right? So it becomes positive. And that actually has sort of a repulsory effect on any sperm that are really close by. It just causes them to bounce back. It's like two positives are going to repel each other, or if you put two magnets together the wrong way, they want to push apart. So that depolarization is produced by a calcium wave, right? Calcium all of a sudden is going to go rushing in, and now the polarity is going to change. And that's triggered by the sperm when the sperm binds. And it happens right at the spot where the sperm binds, and it looks like something from a sci-fi movie where you have like, you know, like a meteor hits a planet and then the thing goes all the way around. It literally does that. It just starts at that one spot where the sperm bound and travels all the way around the egg and knocks any of the rest of the sperm that might be touching the egg off. Pop. They pop away. But this is going to transition back. It's going to change back really quickly. Within a minute, it'll change back to being negative again. Then the sperm could go right back on, right? So you have to make a permanent prevention to keep them from getting in there. And that's known as the slow block, although it's not that slow, okay? It's just slow relative to the depolarization. So within about 5 seconds, the cortical granules release their contents, and those contents form a dense mesh of extracellular fibers of proteins all around the egg, and the sperm have the enzymes that the sperm have are useless on these glycoproteins. So they can't chew their way through them. So that one sperm goes through and that's it, okay? So that's known as the permanent block to polyspermy, because no sperm would get in there. Is it possible that 2 sperm could hit the egg cell at exactly the same time? Yes. Okay, it's unlikely, but it's possible. And what that would do is it would start embryonic development, but very shortly after embryonic development, in this stage right here, in the cleavage stage, the cells would die. And there would be no pregnancy, no implantation, no embryo. It would just those cells would die. So that's not going to happen that you're not going to see that as a consequence, but it does happen. Pardon me. Thank you. So the sperm comes through, it has to work its way through these outer layer of cells called the coronal cells, basically digest away the zona pellucida, create a hole where it can pass through. And once it gets through, it touches the egg membrane, and immediately there's a depolarization. If another sperm was right here, it kind of gets repelled. And then all of these little cortical granules will fuse with the membrane and release their contents, which are going to harden up into essentially another membrane outside the egg. And that is known as the fertilization envelope, but it's impermeable by sperm. So within a few minutes of sperm binding, the sperm itself, essentially the sperm's membrane, cellular membrane, and the egg membrane fuse into just a continuous membrane. And so the nucleus of the sperm cell is now released inside the egg. And that is going to trigger the completion of meiosis II. So remember that the egg was arrested in prophase 2 or metaphase 2, and it has to finish meiosis. And it does it after fertilization. So the egg nucleus over here and the little sperm nucleus that came in don't fuse immediately. So this nucleus is going to have to go ahead and divide again by meiosis. So it's arrested in, if we looked at it, all the chromosomes would be lined up. So it's in metaphase, what we call metaphase 2. And it has to get out of it. It has to finish that. So it does, and that's what's shown happening here, I guess. There's the sperm, and here it is finishing meiosis II and creating another polar body. And that polar body is very, very tiny. It's as small as a sperm, and it's not going to be of any consequence. Then the nucleus of the sperm and the nucleus of the egg, these are now called pronuclei, will be able to merge together and fuse to form a single nucleus. And at that point, we have a true zygote. Okay? So this happens, this is hours after fertilization. This takes probably 4 to 5 or 6 hours before this all happens. Okay? Now, when the sperm enters, the membrane fuses, and the tail was part of that membrane, and the tail had the mitochondria. So the mitochondria never usually gets inside the egg. It usually actually is just lost outside the egg. So that means that all of the mitochondria that an individual inherits came from the egg, which were the exact same mitochondria that the mother had originally. So all of us have mitochondria that we inherited from our mother, who inherited from her mother, who inherited it from her mother, who inherited from her mother. And an interesting thing that people did was to look at the genes that are in the mitochondria, because it has its own set of genes, and track the differences that they have, and essentially look at that for inheritance. So you basically can track your maternal inheritance all the way back through the mitochondria. And this is what some of those places like the 23andMe and Ancestry.com, these are some of the genes that they look at to look at your ancestry. So after the pronuclei fuse and you have a zygote, it immediately starts the process of mitosis. And you're going to have a 2-cell stage, 4-cell stage, 8-cell stage. Those series of cell divisions are going to be very synchronous. And so this is known as the cleavage stage, the cleavage period here. The cell divisions start out, the first couple of them are fairly slow, looking at 15 hours or so, and then they start to speed up. So as we progress, they get a little faster and faster until they're running at about a cell division every 8 hours. So about 3 divisions every day. So after 1 day, you've got this 4-cell embryo. After 4 days, you have about 256 cells. And then after 6 days, you have about 4,096 cells. And you calculate that by just 2 to the n, right? So however many divisions you have, 2 raised to that number of divisions. So at this point, you would have had approximately 12 divisions, and that would be 4,096 cells. When we look at it at about 3 days or so, you start to see that it's formed a cluster of cells, and it sometimes is called a morula stage. Morula means cluster, so it's a cluster of cells. But by the 4th day, if you look at this cluster of cells, if you look inside of it, you see that there's a hollow area in there. Most of the cells are just around the outside surface, but on one side, there will be a cluster of them, a little lump of cells that are there. And that little cluster is known as the inner cell mass. And that, part of that will become you. Okay? Part of it will become stuff that you lose, your part of your placenta, but part of it will become you. Okay? And that inner cell mass will be just on one little edge of that blastula, or as I said, a lot of times it's called blastocyst, because something that's a fluid-filled pocket is known as a cyst, and this is not hollow, it's filled with liquid. So somewhere between day 4 and day 6 is the optimal window for implantation. So if fertilization occurred and the egg was up in the fallopian tubes, you have 2 or 3 days for the egg to for the embryo to start to travel down the fallopian tube and get to the uterus where it can implant. If fertilization happened in the uterus, then it would not be in the right place in the uterus. It would either implant really close to the cervix, which usually ends up being it aborts, or it doesn't get to implant at all. So it has to kind of be at the right place at the right time for fertilization to occur and then subsequent implantation to occur. So implantation is where the embryo contacts the lining, the endometrial lining, and the outer layer of that endometrium is these columnar epithelial cells, and it makes contact with them, and it kind of rolls along. But the cells that are on the side where the inner cell mass is have the ability to adhere. So they will stick. Once they stick, then these cells on this side begin to differentiate. And they do some really weird differentiation stuff. What they do is they start to fuse with one another. Okay? And when two cells fuse and they share their same nuclei, both of their nuclei stay in there, they are now known as a syncytium. And so these cells start to form a syncytium, where they essentially start to merge together, and that's shown here. And they're known as syncytiotrophoblasts. So this entire blastula has at least 3 different types of cells in it at this point. It has the inner cell mass cells, and we could also call those cells embryonic stem cells. And you may have heard of things like stem cell therapy with using cells from frozen embryos. Well, it's these cells from the frozen embryos that are able to be used because they have the ability to generate all different types of cells that will ever exist in the body, right? That's where you came from. So those embryonic stem cells have the complete capacity to be any type of cell in the human body. Okay? The other types of cells are the ones that make contact. So those are going to become the syncytiotrophoblasts. And then the other cells that don't make initial contact are just called trophoblasts. Okay? So the trophoblasts are going to just maintain sort of an outer wall of cells as the syncytiotrophoblasts begin to wedge their way into the endometrial tissue and start to cause changes in the endometrial cells, which are going to lead to the formation of the placenta. So one of those changes is they trigger the capillaries in the endometrium to start forming essentially sinuses. They just start to break down their capillary walls and become just pools of blood contained by the syncytiotrophoblasts cells. And that's going to become the basis for the placenta. Okay, so that's shown here where you can see some color to try to demonstrate the different parts. So these are representing blood vessels that would be there in the capillaries, and the syncytiotrophoblasts, okay, is really one giant cell with a bunch of nuclei in it, and it's invading this tissue, okay? So, you think about movies like Alien, where your body's being taken over by another life form. It's literally taking over the endometrium. It's another life form taking over the endometrium, okay? Now, there are lots of changes happening here with these cells, but the embryo itself is also beginning to develop. Okay, so that inner cell mass starts to form a 2-layer disc of cells. So one layer of those cells are on the endometrial side and facing the syncytiotrophoblasts, and the other layer is going to be facing the blastocele, the hollow space here, that's in the blastula. And so they're in a different environment, and those cells start to differentiate. They start to change. The layer next to the endometrial side becomes what's called the epiblast, epi on top, and the layer that's next to the blastocele, the yellow spot here, the yellow cells, become known as the hypoblast, underneath layer. Okay, epiblast, top layer, hypoblast, bottom layer. So it starts to form this two-layer arrangement, and another hollow space begins to form within that upper layer. And that hollow space is known as the amniotic cavity. Okay. What happens right before women have their baby? The water breaks. Okay. The water is the amniotic fluid. So ultimately, this amniotic cavity will expand to surround the entire developing fetus. Okay, and fill a fluid-filled sac around it that will rupture right before or during birth. Okay, so that starts out as a cavity here in this upper layer. So this is going to be taking place over a period of about a week following implantation, up to about 14 days. You have the process of the embryo expanding into two layers, and then at the end of that time period, that two-layer embryo is going to undergo the process of gastrulation. So implantation occurs while it's blastula. Okay, at that blastula stage, more or less continues through the gastrulation stage. And by that point, you have, this is an embryo that is now at the gastrula stage, and we'll go into more detail about what's happening there, those changes. But you still have this sort of three-layered embryo between two fluid-filled vesicles, okay? One of those vesicles is the amniotic cavity. The other one is the yolk sac. And the yolk sac is nothing but the remnant of the blastocele, okay? So there are, at this stage, one really crucial thing going to happen. Cells that are on the inside of this yolk sac turn loose, and they migrate back into this region and back up into here and take residence in the area that is going to become the urogenital ridge, which will eventually be the gonads. So that's where the embryonic stem cell population of cells come from. It's also where our blood cells, the cells that are going to become the bone marrow, come from. So outside of the embryo are essentially, here's the embryo itself, but outside of the embryo is a lot of tissue that all came from the embryo. Those tissues are called extra-embryonic tissues. And they're what's going to constitute the placenta and these membranes that surround the embryo. Okay, so the membranes that surround the embryo are the chorionic membrane, the amniotic membrane, and the allantoic membrane. Okay, and they form cavities known as the chorion, or the chorionic space, or the amniotic cavity, or the allantoic cavity, also known as the yolk sac. And those membranes will continue to expand and basically form then a three-layer arrangement that is the placenta. So the placenta is the chorionic membrane, and the amniotic membrane will basically just grow into each other. As the amniotic cavity continues to expand, where does it go? Well, it runs into the chorionic membrane, and the allantoic membrane basically just is surrounded within them as a cavity. So these three membranes form what eventually ruptures when the water breaks. The chorionic membrane and the allantoic membrane are giving rise to the interaction, the interface between the endometrium and the embryo, and become the placenta. So the placenta tissue itself basically just expands to take over the entire inner surface of the uterine wall. and the blood vessel exchange there, you have blood coming from the mother, which basically just pours into these venous sinuses, and gaseous exchange can take place over those three membranes to capillaries that are within the chorionic membrane, and those capillaries feed back into the placental vein, which we'll see dumps into the venous return blood flow in the developing embryo. And the embryo's heart is pumping blood out through arteries into capillaries into the placenta. All right, so one other thing that's happening, these syncytial trophoblasts and trophoblast cells begin cranking out a hormone that they make, chorionic gonadotropin, human chorionic gonadotropin, HCG, but you could just call it CG, chorionic gonadotropin. And it is going to basically reproduce the effects of progesterone. It also is mimicking luteinizing hormone, which luteinizing hormone increase the amount of progesterone as well. But what it's doing is it's maintaining the corpus luteum in the ovary, and the corpus luteum is cranking out progesterone. So HCG is essentially making progesterone still be made, and HCG itself acts like progesterone. It's in that effect. And so what does that do? Well, it keeps ovulation from happening again, because you don't need another egg if you're pregnant. And it maintains the endometrial lining and causes it to even continue growing. And it revamps the metabolism in your mother's whole body to be able to mobilize and store fats. Okay. So one thing that goes along with a developing embryo in here is weight gain in the mother, usually in preparation for the production of extra blood and extra nutrients that need to get to that embryo. It will also induce lactation. It'll induce the hypothalamus to stimulate the pituitary to release prolactin, which will stimulate mammary duct proliferation and therefore milk production in the mammary glands. So what happens to these membranes? So the amniotic cavity is ultimately, as I said, going to surround the entire embryo, basically creates a aqueous environment to develop in, sort of acts as shock absorption and so forth. Once the baby gets big enough to be a fetus, it's not so much shock absorption in there. The yolk sac will be incorporated into the gut. So it'll just be incorporated into the body, the gut cavity, as the gut forms when the body forms subsequent to these layers forming right here. So ultimately it'll just become the gut. But it is the source of the cells that become the germ cells and the blood cells, the bone marrow. The actually hematopoietic cells. The allantois is this extension off the yolk sac here, and ultimately it'll become part of the reproductive structure or the urinary tract, and that's where the cells get into the reproductive tract. But it's essentially a waste reservoir. So it becomes this waste reservoir serving the purpose of basically holding nitrogenous wastes where they can be absorbed into the placental blood flow. And it's basically will end up being part of the urinary tract. All right, so let's look at what's happening with the embryo itself in the formation of, as it was implanting, it formed basically a two-layer disc, okay? And if we look at this, one side of that disc is the amniotic membrane. The light blue that goes over the top is the amnion. The thicker blue cells here are the epiblasts. So this is not embryo, this is embryo. This is extra embryonic, this is embryonic. So you don't have any part of the allantois on you when you're born. Okay? So these two layers of the primitive embryo are going to start differentiating with each other in response to each other. They're sending signals back and forth. And the hypoblast cells will induce the epiblast cells to start to divide and delaminate at a particular little region that is known as the primitive streak, because it is a little streak that you can see on the developing embryo. Okay. Now, a lot of these terminologies come from the study of chicken embryos, and that's because if you look at our human embryo and you look at a chicken embryo at this stage, you'd be hard pressed to tell them apart. Okay. If nobody told you where they came from, you'd look at them on a slide, you'd go, well, it's a chicken embryo. Like, no, that's a human embryo. No, well, then that's a human embryo. Nope, that's a chicken embryo. They look exactly the same here, as do all other vertebrate organisms. Okay, so this primitive streak is where cells are going to start to move. And here's what they're doing. The epiblast cells have been telling these hypoblasts, the hypoblasts have been telling these epiblast cells to differentiate. And so they start turning loose and they start moving. And this is the beginning of the gastrulation process. So as cells start to move, what they're ultimately going to form is a layer of cells called the endoderm. And they're just going to invade and move aside the cells of the hypoblast. Okay. A few hypoblast cells remain, but they're basically just going to be displaced by these endoderm cells. Okay, once the endoderm cells have replaced the hypoblast cells, they continue that communication, and the cells continue to stream and delaminate, but now they do not invade this bottom layer. They create themselves a new layer in between them. So they push the two layers apart. They push the hypoblast and the endoderm apart. Okay. When this happens, we change the names. So the hypoblast now is named the ectoderm, the outer layer. The hypoblasts are now termed endoderm, inner layer. And the middle layer of cells that wedges itself in there is known as the mesoderm, the middle layer. So at this process is called gastrulation. This is the process of gastrulation. What's going to form as a consequence of that is a disc of cells that have three layers. They have an outer layer, ectoderm, middle layer, mesoderm, and inner layer, endoderm. And it's almost universally that you see drawings of these, they will be colored ectoderm blue, mesoderm red, endoderm yellow. And I don't know who decided that's the way we're going to color them, but that's the way it's almost universally represented. They don't have any color in real life. They're all the same color. They're all clear, basically. All right, so that is gastrulation. And the gastrulation process, you're like, well, I thought you said that forms the gut. Well, the other thing that happens is the embryo begins to change shape. It begins to fold over. Okay. And as it folds over, it's eventually going to roll into a tube. And the middle of that tube is all endoderm. And that's where your gut forms. Okay. So that's why it's called the gut formation. The place where the cells begin to move in, and we can see this, is a place called the primitive streak. And the top of the primitive streak, or the tip of it here, actually starts towards the posterior end of the embryo and moves in an anterior direction. So what else is happening during gastrulation is establishment of what we will say are the three body axes. So the anterior posterior axis, the left right axis, and the dorsal ventral axis. Okay. So this ectoderm is going to be the dorsal side. So the primitive streak is forming in the precise middle of the dorsal side of the embryo. Okay, now, so what's in the precise middle of your back? Your spine. And this is also where the neural tube is going to form in the next stage of neurulation. But at this stage, it's simply a groove where cells are moving. So the cells that move in here have to come from somewhere. These cells are continuing, all the cells are continuing to divide. So as these cells divide, they push towards the middle, and in the middle, they come off and they go back and push towards the outside. So essentially there's this flow of cells. If you look at that, my hands are the top layer, they're continuing to divide, so they're pushing this way, and then they're just curving under and going underneath, like that, right? They're just continuing to push. Once they go inside, they're called mesoderm. While they're still on the outside, they're ectoderm, okay? Once they become once they go on the inside and become mesoderm, they get different communication signals, okay, and they change. So the mesoderm and the ectoderm and the endoderm will interact with each other, and they'll produce different types of structures and different tissues ultimately in the body. So these are known as germ layers because they're going to give rise to new things. They're going to give rise to new kinds of cells. At this point, we really kind of have three different types of cells making up the embryo. We have ectoderm cells, mesoderm cells, and endoderm cells. And every ectoderm cell looks like every other ectoderm cell, and every mesoderm cell looks like every other mesoderm cell, and so on. But pretty soon, they're going to start to take on new roles. So what happens to these cells after they get on the inside? The mesoderm cells form sort of three clusters. One cluster right in the middle, and then two clusters on either side of that middle, right? And you can see that in that top right picture up here. Okay, they're not showing you the whole embryo. They're kind of looking at a cross section kind of through the embryo here, right? So if we look right in the middle, the cells form sort of a cylindrical rod. The mesoderm cells form this sort of cylindrical rod that runs this whole length. Okay, it looks darker up here in the top, and that rod is called the notochord, and it's kind of the first really super important part of the embryo that forms. That notochord is the source of signals that's going to induce changes to create the central nervous system. Okay? So the notochord is there in the middle, and on either side of it, you have mesoderm on the side, okay, mesoderm, but it's going to form little clusters called somites, okay? So what does that notochord do? It's sending out a signal, and the signal is being received primarily by the overlying ectoderm cells, right, that are right above it. And that signal is going to change those cells. It's going to make them get taller and skinnier, and they keep dividing, but they get taller and skinnier, and they start to bend, okay? So you have a series of cells that are pretty much like little columns, and then they are told to get wide at the bottom and skinny at the top. So you have a notochord that's made up of a bunch of cells, okay? Basically form a little rod. And then you have these tall, skinny cells above them. And what the notochord is going to basically do is say, all right, down here at the bottom, I want you to get wider. So it gets wider. And at the top, I want you to get skinnier. Okay? So that one gets skinnier. Well, the next one next to it gets wider at the bottom and skinnier at the top. And the next one gets wider at the bottom and skinnier at the top. So what starts happening? They start looking like pie wedges, right? That means the cells start doing this. They start bending. And when they start bending, then they push ones up on the edge and they get bent down in the middle and they form a little indentation. That indentation is called the neural groove. And that will start to form all the way down the midline, the back of the embryo. Okay? Now everything is still growing. So the embryo has gotten longer, okay, at this point. And this neural groove starts to form and it keeps on going until basically the edges pop back to the middle and touch. And now what do you have? You have a couple of ridges and a big, essentially hollow valley here. And eventually those two ridges will come in contact with each other and fuse. At that point, you've got a tube. Oh, we're going to call that the neural tube. And that's the beginning of your central nervous system. Okay, in the anterior end of the embryo, it's going to be your head, and in the posterior end and the trunk end, it's going to become your spinal cord. Okay, and during this same period of time, remember I said the embryo was basically flat, and then it's going to start folding on this side. Well, the outside edges, way out from the edges, they're not showing you, they start folding the other way. They start bending this way, right? So you basically are forming, the edges are starting to fold this way, and the top is folding over this way. All along the dorsal midline of the embryo. They actually fuse together first in what's going to become your neck, okay? So if you look and track this back into embryonic history, the first place that they fuse together is going to be right here at the base of your neck, kind of right between your shoulder blades, where you can feel that one good vertebrae right there. That's where it fuses first. Then it zips the rest of the way. It zips up the head and then zips down the tail, posterior of the embryo. And as it fuses together, on either side of the neural tube, the mesoderm starts forming clumps, and those clumps are known as somites, and that's what's going to give rise to your vertebrae, okay? All of the vertebrae all the way down your spinal cord. So you start having those first vertebrae right there in your neck. Then the head part doesn't have vertebrae in it, right? And you keep making the vertebrae as the embryo gets longer, but the head part closes up without forming vertebrae. It forms a cranium, right? So the mesoderm on the part of the head up here is a little different. So the notochord, or the mesoderm, forms three really important sets of structures. The notochord is the first one, and what is it? It's a middle cord of cells, like a rod, that go all the way up and down the length of the body in the midline and induce, right, cause to form the neural groove and set about the process of neurulation, okay? Well, how far along are we here? We're at about the 3rd week of pregnancy. We're at about 20 days when neurulation is really underway, and the neural tube generally starts to close at day 21, 3 weeks, okay? So implantation has been there. It's been implanted for about 2 weeks, okay? 3 weeks. When do people first usually notice that they might be pregnant? How long does that take? 4 to 5 weeks. 4 to 5 weeks, right? So at this point, you're pregnant and you have no idea. So this is why they say, you know, don't drink if you are trying to get pregnant, because it's at this stage where a lot of chemicals would have very dramatic effects on embryonic development. Caffeine maybe has an effect. Oddly enough, caffeine in cells in certain studies actually enhances the ability of cells to differentiate into the neural pathway. So maybe people shouldn't lay off the caffeine because those little babies need more neural system. But the other things like alcohol and other chemicals and, you know, any sort of drugs that the mother is taking can exert effects on the cells here. And really, the ones that cause the most problems are the ones that slow cell division. So alcohol is toxic and slows cell division. Anything that decreases oxygen exchange, so that's why smoking or being in a smoky environment or sitting around a campfire and breathing in a lot of smoke or being in a low oxygen environment, things like that, any of that stuff can have somewhat of a deleterious effect on cell division rates because you're just decreasing oxygen that those cells need for metabolism. So anything that slows this process down in here could be detrimental to it. Anything that could cause cytotoxic things, lack of enough certain vitamins to maintain cell divisions, which is what they say prenatal vitamins are important. One of the main ones to take are some of the B vitamins. Folic acid, for example, is really correlated with failure of the neural tube to close. So failure of the neural tube to close properly causes something called spina bifida, which is a baby is born with a not fully closed spinal column. So essentially their spinal column can't develop if it doesn't close. So part of their spine is not functioning properly. The nerves won't develop properly. So those can all be problematic things. So that's why these first 3 weeks, especially the third and fourth weeks of the second, third, and fourth weeks of embryonic development, gastrulation, neurulation, and the onset of organogenesis are really, really critical window of time for the embryo. Okay? Once you have a fetus, whatever, occasional beer probably won't hurt. Because at that point, cell division is not super fast. It's more or less at the same rate as we would have. And the critical parts have all been formed. Okay. So by 5 weeks, 6 weeks of embryonic development, you've basically laid down the foundations for everything that the embryo is going to have. At that point, it's just increase in size, get bigger. So the process of cells changing their fate and becoming different is called differentiation. So I just want to mention that. So these primary germ layers are going to differentiate into specific sorts of cell types in the body. And we look at the embryonic origin of a lot of cell types. Okay, so when we start out talking in physics and A&P, right? One of the early things you talk about in the first semester is histology and tissue types, right? So can y'all just, if you remember, can you remind me the big tissue types? Connective. Okay, and epithelial. Muscle. And neural. Okay, blood, connective. Cartilage, bone, blood, connective. Okay, I want to change the order that these are written down in. Because now we can put them in their embryonic origin. Meso and ecto, okay? The endoderm gives rise to epithelial cell types as well. But the endoderm will make transitions to be epithelial, as well as some tissues that will become mesodermal-ish that give rise to the glands, and like the liver and the pancreas and the spleen, all that stuff, okay? Neural tissue is straight up ectoderm. and epithelial cells are straight up ectoderm. Muscle cells and connective cells originate from the mesoderm. Okay. The endoderm just sort of joins in. Okay. It becomes mostly epithelial in type, but it's going to be the epithelium of places like in the gut. Okay. Now, what about the blood? Blood is a connective tissue. Well, blood actually derives from mesodermal cell types. The cells of the blood, the stem cells, derive from outside the embryo. Remember, they came from the yolk sac. So they're weird. They come in. And they are a permanent stem cell population. The hematopoietic cells are permanently stem cells. They never fully differentiate, right? Muscle cells fully differentiate into muscles, and the number of stem cells for muscle development plummets during early embryogenesis. So you have some stem cell populations that sort of start out from outside the embryo and set up shop somewhere inside the body, like in the bone marrow or in the gonads. Guess what? Those stem cell populations never do anything except be stem cell populations either. Okay, so the gamete or the germ cell stem cells also came from the yolk sac. And this is kind of showing you how they get in. So they come in this way. They migrate into the mesodermal ridge right here. This is known as the urogenital ridge in the mesoderm. This little, it's not a ridge right now in this drawing, okay, but here it is. That's the urogenital ridge. So that structure will give rise to the kidney and the gonads in sequence, right? We talked about last time. Those are unique cells, and they come from the yolk sac. And so is our bone, our hematopoietic stem cells. They're unique cells, actually. They come from the bone. Now, blood is thrown into connective tissues because it has extracellular matrix. That's really a weird way to put it in there. The blood itself is a connective tissue, but the blood cells aren't derived from that same starting point. So all of the cell layers we can track to how they interact and become these different kinds of things. So, for example, in looking at what changes happens to them, they undergo what we call inductive events. So an inductive event is when one cell layer can talk to another cell layer. So, for example, the mesoderm, the early mesoderm, the notochord, for example, sends signals to the ectoderm and causes it to change into neural tissue. And now you have a new layer there temporarily known as the neuroectoderm. And so you have kind of two layers. You have a layer that's the ectoderm here, and you have a layer underneath it that's the neuroectoderm. And guess what they do? The neuroectoderm sends signals to the ectoderm again. Okay, so in one particular place, this is what forms your eye. So you have the neural tube, right? Here it is, and it's surrounded by ectoderm. Well, in the part up here by your head, where your head's developing, they're in contact, and it sends a signal and says, hey, you're going to become the retina. And then the retina, another neural layer, sends a signal that says, okay, you're going to become the cornea and the lens to the ectoderm. And eventually that becomes the eye. And the same thing happens in the inner ear to form the cochlea and then the tympanic membranes in your inner ear. So these interactions continue throughout the rest of the organogenesis process, where essentially you look at and can trace back all of our structures in our body to some series of interactions between germ layers. So between mesoderm and ectoderm, or mesoderm and ectoderm and endoderm, which is making up things like the liver and the pancreas, You have the endoderm layer there, and the mesoderm is in between there and it interacts. The lung, that's an interaction between endoderm and ectoderm to form the lungs and the respiratory tract. So all of these germ layers, the reason we use the term germ layer is it gives rise, germinates, gives rise to these other tissue types. As those tissues form, that process is known as differentiation. Okay, so it requires cell signaling. And I know we haven't gone into cell signaling and things, but there's a lot of cool growth factors that are being produced. For example, they have some cool names, like this particular growth factor here. It's called Sonic Hedgehog and things like that. They're named after weird characters and things like that, but they are growth factors that are produced. You also have the development of the arms and legs that are outgrowths. They're induced by the mesoderm talking to the ectoderm. So there's all these communications that are going on during the organogenesis period. So these slides kind of reiterate what I'm saying there. The mesoderm, what does it give rise to? The endoderm, what does it give rise to? And the ectoderm, which gives rise to the nervous system. I don't guess it was on a slide by itself, but the ectoderm maybe is the most important one. So I mentioned that the embryo does this wrapping around thing, and that process is known as folding. And as the membranes start out, right, you basically have two spheres. One's the amniotic cavity sphere, and one's the yolk sac sphere. And essentially, this one's going to expand while this one doesn't. And so it's going to grow out around it and eventually just fold into it, fold in on all sides. As it folds this way, it's the head fold. As it folds this way, it's the tail fold. And on each side, it's the lateral folds. But essentially, you have an embryo, and those membranes here essentially will continue to shrink as the rest of the embryo grows, and this becomes the umbilical cord. And there's going to be blood vessels that start to form there, and those cells form from cells from the yolk sac. The cells invade and form the reproductive cells and so forth. All during this folding stage is happening somewhere around that 20th, 22nd day, along with the process of neurulation. So some of the key things that are different between embryos and adults, and we'll talk about their circulatory system. So let's take a break, and then we'll come back and we'll talk about that and the pregnancy itself and the changes that happen there.
And then they scan every time if you need to they scan you on your way there. Yeah.
I guess I can get a beer. I think you would hear it. Sexually like that. Yes, that. But even still, like, well, how do I know what to write?
I don't know. The recording your own
Yes, you have to read it out loud. Did you just put it all on?
Yeah, but I can find it.
I really didn't work it all out.
She was
Excuse me, how long is it? What if I take it? Should I take that? Can you pick what section?
So, yeah, so I'll take that. Sure, I was like, I'll make it back for you.
It's really yeah.
You think chemistry is the only take?
So I just have to get above this. Yeah, one more.
So it's supposed to be
What'd you do? Good. Yeah, she's not so much have 80s.
Yeah.
And it wasn't the ones you took, which is make grammar, math, reading, okay. Just release it. What did you make? Like, did you make above a 70 on all of the sections? The HESI?
No, no. No, we made like 68.
Oh.
But you have to make a 70 overall.
I thought it was just like each section had like a 70.
But I think it's overall. They'll give you like an overall score after you.
Okay, so I wrote it. Yeah. Just talk about myself. That's right. Did you?
Yeah, I got it, but I'm not too. I am not doing.
That's like highway robbery. You know how much they charge for everything else? Clothes, hair, and that.
She called me yesterday, she was like, I'm going to my person. Thank you, but no thank you. which was, I was, and then when I came here, we didn't have any last week. And I didn't have any. Yeah, like minimum. I will tell. Like, you're gonna be rude, yeah. No, I would like love things to work different. Yeah, I'm not gonna be there for you. Yeah, you don't want to be there for a year and then finally go, here's the holidays. There's other jobs. Yeah.
Do you think we have to have our background checks stuff done before this fall?
I'm gonna go ahead, like, I'm about to go print out my application stuff, and I guess I'll just do it after like next week or something. Just so it's done.
That's what I do, so I like mine before my name. But then, they want our transcript on your way.
Yeah.
But then this policy, where we get numbers, policies.
Like, I'm not gonna get this transcript, it's not great. Because it's not only two. She goes,
Well, the background check, or is it after we're accepted and we do the spring semester and then that? Yeah, because the fall semester and then,
I know we don't have to be before fall, I'm sure we just have to have it done before like spring or something.
I don't know, it says before the first fall semester.
Or maybe we have until
Yeah.
next fall, because it starts
That's on.
because
I don't see
can't
them
expect
doing,
us to do all that and then risk not getting.
yeah, I don't think they would make us. Talking about.
If it seems illogical, it's almost certainly what they want.
Yeah.
All right, so we're going to look at the blood vessel circulatory system that is very different in the developing fetus. Since it's not using lungs to get oxygenated blood, it's got to get the oxygen coming from the placental exchange. So how does that blood get into the circulatory system? And then what happens to it after you're born? So the placental vein, which is very large, coming in from the placenta, bringing oxygenated blood. Okay, so remember that red is usually meaning oxygenated, pink is less oxygenated, blue is low oxygenated, and so forth. So the oxygenated blood, the highest oxygenated blood that there will be here, is coming in, and it gets shunted straight into the vena cava, just behind the liver, in what's known as the ductus venosus vessel. So this little shunt across here goes into the vena cava, the blood flow of all of the systemic circulation coming back to the heart. The heart pumps it. And so normally in adult circulation, the heart would pump from the right ventricle to the lungs. Well, the lungs are not doing any function for respiration right now because the embryo is surrounded by amniotic fluid, and the lungs, in fact, are filled with amniotic fluid. And at this point, at 3.5 weeks, you don't have alveoli. You don't have all of the structure of the lung anyway. So that blood is not really doing any oxygenating potential over here. So most of it, a good percentage of it, is bypassing the lungs entirely through the structure known as the ductus arteriosus. More of it is bypassing the right ventricle entirely by traveling straight through an opening between the two atria. It's really a flap-like opening. It sort of has a flap that bends open like that and folds back across itself, but it's called the foramen ovale, okay? So some of the blood is coming up the vena cava into the backside of the atrium and then right on through the backside of the atrium from the right atrium over into the left atrium, where it's then going to go into the left ventricle. And all of that blood is going to be pumped out and go out to the systemic circulation. All right, so now the systemic circulation needs to send blood back to the placenta. So the arteries that go back to the placenta are coming off or branch off just around after the iliosacral branch in the lower abdominal tract. Two arteries, they branch off, they go kind of behind the bladder, the developing bladder, which at this point early on, right, was also the allantoic connection to the allantois, which is out here, and go along that and come out of the body cavity and then into the umbilical cord and then into the placenta, okay? So the circulatory route is most oxygenated blood coming in through the placental vein, okay, via the umbilical cord, comes in, merges with the vena cava behind the liver through a structure called the ductus venosus, goes into the heart. Some of it passes through the foramen ovale from right atrium to left atrium. Some of it from the right atrium goes into the pulmonary trunk, where it's then diverted through the ductus arteriosus straight into the aorta. So largely the blood, probably 75% of it or 80% of it bypasses the pulmonary side of the circuit of the loop, okay, and goes on to the body, bringing moderately oxygenated blood on to the body. All right, so what happens to all of those things after pregnancy? Where is that slide? All right, so after pregnancy, the umbilical cord is pinched shut, okay? They clamp that off, and then it's cut. So what happens to these vessels? Well, there is zero blood flow back through that arterial vein, that umbilical vein. So it just collapses. The arteries are still getting blood flow, but it can't go anywhere. So they harden and they fibrose over a period of weeks. So these become ligaments, okay, become known as the umbilical ligaments. And they basically sort of attach the bladder to the front side of the body wall, your abdominal cavity here. And this becomes a ligament known as the ligamentum teres, or the round ligament. And the ductus, the ductus venosa that would have been going across here into that from the liver, it becomes sort of part of the septum that divides the liver lobes. The foramen ovale, since there's essentially much less blood flow coming up the vena cava from this vessel, that foramen ovale is pushed shut. And it's also pushed shut because the blood pressure coming from the superior vena cava is greater. And the blood pressure through the aorta is greater, and the blood pressure through the pulmonary trunk is greater. So that all pushes that valve, that foramen ovale, closed. It doesn't seal it, it pushes it closed. And in some people, it doesn't quite close, and that can be known as a congenital murmur, where you have some flow from one chamber to the other chamber. But that would close with blood flow coming, more blood flow coming straight through the pulmonary artery now, and resistance in the lungs, because now there's oxygen, air in the alveoli, The resistance here means most of that blood does not flow through the ductus arteriosus. So it constricts. Also, the band of muscle there is sensitive to oxytocin. And oxytocin is being highly secreted during the birthing process. And that helps to constrict all of these arteries and help to shunt blood flow away from them. So that will become the ligamentum arteriosum, which kind of connects the pulmonary artery to the aorta. So all those changes need to happen, and they happen just shortly after birth. So sometimes they don't happen completely real quickly. And so babies are always coded with, like, you know, sort of a number system of how oxygenated do they seem, right? They'll put the little pulse ox on there, and like you'll be 98, 99, 97%. They're usually not quite that high right after they're born, because a lot of the blood is not going to the lungs. It's still going like straight from this side through the aorta and not going all the way through the lungs. So over a period of a few hours or days, their color improves and they get a little bit more oxygenated. All right, so up through about 9 weeks, we kind of say that that's the embryonic period. From 9 weeks on to the rest of gestation, which is 38 weeks, which is not 9 months, 9.5 months at minimum. So human gestation is considered pretty much good at 38 weeks, but typically goes 39 weeks and is often extended to 40 weeks. Okay? 40 divided by 4 is what? And how many weeks are in a month? 4 usually. So that's really more like 10 months. But, you know, give them that 1 extra month of hope that it's only going to last 9 months. So that gestation period of 38, 39 weeks, the last 2/3 of that basically are the fetal period. Baby's just growing. Things are changing, but all of the parts are essentially laid down from the organ primordia. And they're just simply going to increase in size and functionality. And of course, the growth of the embryo inside the uterus means that the uterus and the mother have to respond to that growth physiologically as well. So there's anatomical changes, obviously, metabolic changes as well, and hormones are inducing those metabolic changes and the physiological changes that have to respond. So your homeostatic balance is being thrown off. You have extra oxygen demand, extra nutrient demand, extra waste removal demand that's happening as a consequence of the fetal development that's going on. So obviously the uterus is going to need to expand. It does it both by being able to stretch, but also by cellular proliferation in the myometrium and the epithelial layers of the uterus. So it enlarges quite a bit. It's going to be increasing in the upward direction because it's bounded by the bony floor of the pelvis in the posterior direction. That's going to push everything else up and back. So there's pressure that's put on the bladder, pressure that's put on the digestive tract, the liver, the diaphragm, makes it increasingly harder to take deep breaths, and so on and so forth. So the HCG, one of the other things that it's doing is inducing changes in hormonal sensitivity. So all of the cartilaginous symphysis connections in the body alter. They become softer or more pliant. Specifically, one in particular is the pubic symphysis, which is the cartilaginous joint between the front of the pubic bone there in the pelvis, right in here. so that can actually bend out and expand, which it's going to need to do. Partly that helps give more room for the uterus to expand, but it also is going to allow for the baby to pass through the vagina to be born. But all of the other cartilage joints in the body are also sensitive to this. So the cartilaginous joints at the rib to the sternum and other places in the body become a little bit more pliant, which is just a consequence of this. This pubic symphysis is the one that needs to become pliant, but the others can become pliant as well. Weight gain, normally 20 pounds, 20, 30 pounds. Some people a little bit more, some people a little bit less. My daughter, or my stepdaughter, I don't know how much she gained, but she doesn't look like she ever had a child, let alone 2 children. So some people don't gain as much, and that can be problematic because the hormone levels that are being produced are kind of having their effect based on the dilution into the body weight mass. So if you don't gain as much weight, they can have an increased effect. So the placenta is making a bunch of hormones, HCG, relaxin, lactogen, and some of its effects are to increase the metabolism, what's called glucose sparing effect. This increases the ability to burn fat, okay? And the increased estrogen and progesterone that's being produced, so the corpus luteum is just cranking out progesterone like crazy, all of that promotes mammary gland cell proliferation. It also induces prolactin, which will induce those epithelial cells to start synthesis of milk. Why do you get morning sickness? Well, usually it's due to those elevated levels of estrogen and progesterone that are happening. They're going to be affecting epithelial proliferation. Well, guess what other cells are epithelial cells? The digestive tract. And they over proliferate to some extent. And that can be a source of nausea. All of the physical displacement of organs is going to cause things like heartburn. Now, morning sickness usually shows up earlier. That's like one of the first symptoms in the movies, right? Well, it is usually going to happen starting around the 5th or 6th week of pregnancy, and it can last the entire pregnancy, but it may usually be worse in the first trimester, the first 3 months. Heartburn and things like that are probably not going to be happening until later, until the uterus is expanded to a point where it's starting to cause pressure on the stomach and the diaphragm and the esophagus, and then pressure on things like the colon and the rectum can cause problems with constipation and motility just in general. Blood pressure will go up. Why? Well, you're pressing on some of the major vessels in your body, right? The aorta and the iliac vessels are being pressed upon, but you're also increasing body mass, which means the heart has to pump harder, which means the blood pressure is going to go up. Respiration rate can go up and heart rate can go up. All right, so 38 weeks, 39 weeks. The way they predict it usually is to say it's going to be within 15 days of a certain date, and they calculate it based off the last recorded menstrual period. There can actually be menstrual flow during implantation. Some of the endometrium may slough off. So sometimes it's miscalculated by 28 days. Okay, so it can be a little off. All right, got this baby. It's reached maturity. It's going to be born. You're going to have the birth, which is parturition, which is a big fancy name for having a baby. Hence the term postpartum depression. It's not after you left, it's after the baby left. And the initiation of labor is typically a factor of the peak level of estrogen and progesterone. So as
pregnancy continues, estrogen and progesterone levels just keep going up. They never peak out. And eventually that's going to reach a point where the expansion relaxation response or reflex that smooth muscle has, it just can't relax anymore. It will not be able to not contract, okay? And these myometrial cells are sensitive to oxytocin, which is a posterior pituitary hormone, remember? Oxytocin is being synthesized by the posterior pituitary because it's being triggered by ACTH, which is being made by the adrenal glands of the fetus. So the mother's body starts to get this increasing dose of ACTH as the adrenal glands mature. And they're one of the last glands to mature in the developing embryo. So the last thing to mature functionality is the lung and the adrenal gland. Well, the adrenal gland is cranking out ACTH, and that induces the pituitary gland to release more oxytocin and more oxytocin and more oxytocin. And that'll cause these smooth muscles to want to contract, but progesterone will sort of prevent that from happening until it reaches a point where, we're just not going to be able to relax anymore, and contractions really start. And that starts the wave-like repeated waves of contractions that the uterine muscle cells will undergo. And it will cause the further relaxation of the cervix as it dilates so that the baby can leave. So stage 1 is the beginning of dilation, and usually the baby has to move around, which it will either be that way from the beginning or it'll get moved around. So it's head down. The head's the heaviest part, so it generally just gravitates that direction anyway. And as this relaxation occurs here and the dilation begins, the allantoic membrane is stretched across that opening, and the baby's head usually pops it. And that's the water breaking. And it might be a whole lot, or it might not be, because as soon as it breaks, the baby's head maybe just seals it right off. Okay, so just a little bit. So a lot of times women think, oh, I just leaked a little bit, because that's already happening. Incontinence is already happening because you've relaxed all of these cartilaginous connections to the muscles of the urethral sphincters, and the bladder is being squished like a clam. So everything is getting pushed, and incontinence, maternity incontinence is pretty common. So yeah, I just peed a little bit. Nope, that was your water breaking. Okay, so once that all starts getting into motion, once the water breaks, generally the baby needs to be born within 24 hours. Stage 2 is the strong muscular contractions that are going to actually expel the baby. So that's the actual birthing process. And then once the baby's out, the umbilical cord will be cut. Well, it doesn't have to be cut, okay? So what used to happen before the dads were allowed into the operating rooms to be able to cut the umbilical cords, okay? When we were still living in caves around campfires. Well, the baby comes out, somebody's holding it, but then the afterbirth or the placenta is delivered. The muscular contractions continue until the blood vessels that are surrounding the placental blood flow essentially constrict, and the placenta will turn loose from the endometrial lining will shed all at once as the placenta, and that can cause a lot of bleeding and hemorrhaging. And if that is ruptured before it's actually through muscular contractions, that could be problematic. Generally, this takes place maybe 20 to 30 minutes post the baby coming out, okay? So the baby comes out, but mom's not done for a little while, okay? Things that are typically done are things like an epidural, which is essentially a sedative or a numbing agent injected straight into the perispinal fluid space that numbs everything, cuts all sensations from that point down. But it doesn't just block sensory impulses, it blocks motor impulses as well. And that slows down the body's ability to induce the person's sort of voluntary control of muscles. It doesn't stop the uterine contractions because they're involuntary contractions, but it makes it harder for the mother to actually do contractions or to breathe and create pressure in the abdominal cavity and those sorts of things. And it will slow down all the events. So that will prolong labor. It will prolong the placental delivery as well. The trade-off is you don't feel it. So you're in there for longer, but you're not feeling it for longer. All right, so we already mentioned this. The fetal circulation has to change so that the fetal blood supply is bringing oxygenated blood from the placenta, and that's not there anymore. So it has to change to bring oxygen from the lungs. So the pulmonary circuit has to lose its connectivity to the systemic circuit, which is this ligamentum arteriosum, which is sensitive to oxytocin as well. So during the birth process, that oxytocin is squeezing that off or causing that smooth muscle to contract and make that change. All those slides just have to do with that same stuff. All right, so now you're a baby and you're hungry. So what are you going to eat? Well, that's where nature has provided mammals with the ability to produce their own nutrition for their offspring via the mammary glands. And those glands have been, epithelium has been triggered to grow from HCG and progestin and the other hormones that the placenta is making. And prolactin has stimulated them to actually produce milk. So now it just has to be delivered to the baby. Well, the oxytocin that's present at the time of birth triggers smooth muscle contractions in and around the mammary cell or the mammary glands as well. And stimulation when the baby nurses, stimulation on the nipple actually releases oxytocin again. And that causes what's known as the milk letdown reflex or response. So it becomes Pavlovian, sort of, so to speak. Even if the baby cries, that automatically sends in motion that milk letdown release, and the muscles will contract and the milk will be secreted, okay, even if the baby's not there yet. So initially, the liquid that's produced by the mammary epithelial cells is a fluid called colostrum, and it is lower in fat and higher in carbohydrates, and also is very rich in antibodies, and that's usually for the first couple of days. Well, it's lower in fat because the baby doesn't have much in the way of enzymatic system going yet for digestion. It's never eaten anything, right? Its stomach is going to be filled with epithelial cells that have been discarded because the epithelial lining is still being replaced in the digestive tract. So there's all this material in there that the baby is going to poop out for the first couple of days. It's basically mostly just dead epithelial cells. So from this colostrum, they get bacteria, the necessary microorganisms that come in to their digestive tract. They get antibodies, and they basically just get a light meal, okay? Not super heavy in fats, a little glucose that can be easily absorbed and provide energy. Eventually, it starts to make more protein and lipid in the milk, and at that point, it's bona fide milk. It has lactalbumin and casein, which are the two main milk proteins, and lipids. And the advantages that it has is those antibodies that are being able to be transmitted from the mother to provide passive immunity to the newborn. The nutrient composition change over time is perfect for our digestive tracts and our growth. There are a number of reasons why nursing might not be able to occur. If the mother is on any kind of medication, that too will go through the breast milk. And so if there's something that the mother has or has to have that the baby cannot be given, then breastfeeding is not going to work. If the baby has a nutritional need or deficiency, for example, the genetic disorder called PKU or phenylketonuria is the inability to break down one of the amino acids, tryptophan, which is going to be present in the proteins of the breast milk. So you want to limit that protein or that amino acid from the baby's diet. So the baby's not going to be able to get breast milk because if that amino acid isn't broken down, it becomes toxic. So you have to make sure you give them a diet that doesn't have that particular amino acid in it, or very, very low amounts of it. So those kinds of conditions are checked for before the baby ever has a chance to eat, so they know what needs to be done. Nah, I don't want to talk about that. Well, if people are not able to have a baby, what we call that, maybe they're infertile or sterile. Sterile means unable to make gametes, actually. But people want to have babies or want to have offspring, want to have kids for whatever reason, and that's fine. So technologies have existed for a long time to try to assist with that. You have to figure out what the problem is to be able to come up with what strategy is going to work to solve it. So looking at the reproductive tract to say, okay, looking at all the processes actually, so step one, are gametes being made? Is there a genetic problem? Is there a reason why gametes might not be being made enough? Like, does one of the parents have a rearrangement in their chromosomes that causes very few gametes to be viable? Or if gametes aren't being produced, is there a problem with maybe the gonads? Has there been trauma? Is there elevated temperature? Is there lack of blood flow? Is there some immune response, autoimmunity, or something like that? Numerous different reasons that gametes might not be being produced. And that's usually on the side of the male, because the eggs were made originally as that 6 weeks old embryo in the female. So it's not like they either had them or they didn't have them. Now, can they get out? Is there a blockage in the reproductive tract? Maybe, right? So there could be blockage in the, there could be scar tissue around the ovary. There could be scar tissue in the opening of the fallopian tube. The fallopian tubes could be damaged. The uterus could be damaged and can't maintain an endometrial lining. There's just a bazillion different things that could be going wrong. Well, if the reproductive tract checks out, it's like, okay, everything should work. Everything could go through there. The sperm could get in, the egg can get out, and there's just not enough sperm. Okay, try to increase sperm count. Ejaculate a whole bunch of times and then save the sperm and then use them all at once. Okay, so if there wasn't enough sperm, if the sperm count was too low, well, just combine a bunch more sperm. If the egg can't get out, take the egg out from the ovary, fertilize it in vitro, and then put it back in. That'll work. Kind of circumvent the blockage. Okay. If eggs aren't being produced, or if ovulation isn't happening, there are certain drugs that can be taken to sort of assist or enhance the ability of ovulation to occur. These are sometimes known as fertility meds. And this has been some of the reasons why some of the people, I remember back in the late 90s, I guess, there was people that had 8 kids, octuplets, yeah. And that was due to taking a drug called Clomid, which is a estrogen inhibitor, or testosterone inhibitor, which makes the effects of estrogen more effective, which means made FSH more effective.
FSH, and they ovulated more than normal. Okay, so normally what they would do is cause ovulation like that, collect the eggs, fertilize them in vitro, and only implant a couple of them. But this got fertilized in vivo, and all 8, or at least 8, there could have been more than 8, but 8 implanted. That's not typical. So these are known as assisted reproductive technology, okay? Now, if assisted reproductive technology takes the tract of inducing super ovulation, making ovulating a bunch of eggs, and those are collected, what do you do with the ones that you don't implant? If you make embryos out of them, what do you do with the ones you don't implant? Can you save embryos? Can you save eggs? Yes, those cells can be frozen and thawed, and then most of the time they work. So a lot of times their eggs are going to be fertilized. The embryo is going to go to that early stage of development. In that cleavage stage, when there's at least 8 cells, 1 cell can be harvested. And you don't take a cell out, you stick a needle into it and suck the nucleus out, and its genes can be analyzed by a variety of means to see if everything is okay genetically. At this point, they can just sequence the whole genome. If everything checks out, then that 7-cell embryo can be implanted. Okay? If it doesn't check out, the 7-cell embryo can be trashed. You're like, well, you can't just throw it away. It's alive. Okay, we'll freeze it. Okay. Yeah, it's alive. So are a lot of the skin cells you lose every day in the shower. Okay. So are a lot of the cells you poop down the toilet. Okay, they're alive. But there's a big ethical dilemma with getting rid of the embryo cell. So freeze it. Now what do you do? Who owns it? Who has to pay embryo support? Okay. So there's a lot of questions ethically about what you do with those cells. Well, most of the time people sign them over and say, well, okay, I'm not going to come back to the freezer every day and pay the electricity bill for this, so they're yours. And they become the property of the assisted reproductive technology company that helped them, or the hospital, or whoever it was, the clinic. And what are they going to do with it? Well, they're going to do research. They're going to take those cells and figure out how they work. And so that's what led to things like embryonic stem cell therapies, which they would say, oh, well, that's cloning. Well, that's just because you're making a whole bunch of identical cells. Yes, that's cloning. You're not making a whole bunch of identical people. So it's not cloning like in the movies, but it's cloning like in real life, which is just making a bunch of copies of cells. So all of that technology, started out with good intentions, like, hey, we just want to help people have babies, to being like, well, now what? And then lawyers get involved, and ethic battles start happening, and then, you're the devil, no, you're the devil, and everybody hates everybody. So we should have stopped it all by saying, yeah, you just can't have a kid. Go home, watch TV. So how do you even get classified as being infertile? Well, technically, you have to have tried to have a kid regular way for a year before you can qualify for assisted reproductive technologies of any kind. And then you can start getting tested for what's really wrong. It's an expensive process that many insurances would never cover in the beginning. Now I think they may cover certain aspects of it, but basically they said not having a kid is not an emergency procedure. It's not a procedure. We're not going to pay you to make more things for us to have to take care of. So insurance companies weren't going to, they're on the bottom line money, and they don't care about your feelings about wanting to have a kid. So they just wouldn't pay for any of that. So it was all not regulated. A lot of it was not regulated. So now you're sort of on the back alleys
doing strange reproductive techniques. There's a lot of shysters out there taking advantage of a lot of people. All right, so tomorrow we'll review for our test. So it's going to cover, where do we go? The digestive tract, right? That's where we started. And the reproductive urogenital tract, the urinary part, but the reproductive part of it. And this the stuff we talked about today. So go and read all of this stuff, read over the notes, come in here tomorrow with lots of questions. I know we covered a bit of stuff kind of fast today, but come in with a lot of questions and we'll figure it all out, all right? And study for your lab test too, which is tomorrow, right?
When I was reading about the digestive system,
Uh-huh.
I found this thing about the Alex St. Martin, what his name. Dude had like a stomach, a hole in his stomach.
Uh-huh.
And the guy was like putting meat in his stomach.
Uh-huh.
That's crazy.
Yep. Yeah, they won't